Okay, gentlemen, today is Wednesday, April the 8th, and today we are going to talk about the fifth and final section of chapter 15. Um, some inheritance patterns are exceptions to the standard Mendelian inheritance. Okay, now, um, genetics is a very complex subject. All right, as a matter of fact, college biology students who take genetics actually spend an entire semester, sometimes two, studying the things that we've covered over the last 4 or 5 days. Right, and so there's a lot of detail and intricacy in these things that we've obviously kind of glossed over a little bit. But this again is a general biology class, not a genetics class. So what does this mean? All right. Well, there are two very normal circumstances in which Mendelian genetics simply don't hold up. The first is an exception that involves genes that are located inside the nucleus. All right, and the other is something that we seldom think about, but there are genes in our bodies that are found outside of the nucleus, not your standard nuclear genes. And in both cases, the gender of the parent contributing an allele is a factor in the inheritance pattern. So let me give you this example. Um, so uh, this is an example that we talk about in your textbook. So you can go through and read specifically about this gene and what it does and that sort of thing. But this is a phenomenon called genomic imprinting. All right. Now, um, what happens is that in mice, a normal allele is found on both the paternal chromosome and the maternal chromosome, but the mother's chromosome is not expressed, and so it doesn't really matter what the genotype is. The offspring's phenotype will be based on whatever allele is found on the paternal chromosome. So let's say that you're a homozygous dominant individual, with the wild type being the dominant allele. Well, in this case, the allele on dad's chromosome gives rise to a normal-sized mouse. On the other hand, if a heterozygous mouse is produced, and here we have a normal allele on dad's chromosome and a mutant allele on mom's chromosome, well, mom's chromosome is not expressed, and therefore the offspring will be a normal-sized mouse. On the other hand, this version of a heterozygote, where the mutant allele is on dad's chromosome and the normal allele is on mom's, in this case, again, the mother's chromosome is not expressed, and therefore the offspring will be a dwarf mouse. Okay. So genomic imprinting involves silencing certain genes depending upon which parent passes them on. And in this example, of course, it's the mother's gene that is silenced. So it seems that imprinting is the result of what we call methylation, which is when we take methyl groups, a carbon with 3 hydrogens, and bond it to cysteine nucleotides, the C in the DNA. Remember A, T, C, and G? So when we methylate those cysteine nucleotides, the gene becomes inactive. It's silenced. And although we're not absolutely certain, it's believed that genomic imprinting only affects a small fraction of genes, and most of them are important in developing for embryos. Now, there's also the fact that some DNA is actually found in organelles called plastids, and also in mitochondria. Now, all organisms, except for the prokaryotes and the bacteria, have mitochondria in them. And the mitochondria actually contains its own DNA. We talked about that when we were talking about the development or the evolutionary development of mitochondria. And so these are called extranuclear genes, or sometimes referred to as cytoplasmic genes. And these are found not only in mitochondria, but in plants, they're also found in chloroplasts. Now, these extranuclear genes are inherited only from the mother, all right, because the mitochondria of sperm in both mammals and animals and plants are not embedded inside the mother's egg when the fertilization event takes place. So you only get your mitochondrial DNA from mom, all right? And this is a picture of a plant where the patches of color that you find on the leaves of the plant are dependent upon the genes that are found in the mitochondria, or in this case, in the chloroplasts. All right, and that's what makes them not be green. They're this bright pink or even brownish color. Now, just as a general summary, some defects in mitochondrial genes can keep the cells from making enough ATP. All right, because remember, the mitochondria is the powerhouse of the cell. And so that can cause diseases that affect your muscles and your nerves. But it may be possible to avoid passing on mitochondrial disorders from mom to offspring if you remove the nucleus of a mother's egg and place it in another woman's egg with healthy mitochondria. And so this egg can then be fertilized by the sperm of the prospective father, and that egg can be transplanted into the womb of the prospective mother. That child will then be genetically the offspring of that mother and father. Okay. All right, fellas. So that brings us to the end of section 15.5 and the end of chapter 15. There is no class tomorrow or Friday, and of course, not all next week. So enjoy the rest of Holy Week. Happy Easter, and I will see you again and talk to you again on Monday, April the 20th.